Hi, my name is Dr. Rita Nanda, and I'm a breast medical oncologist from the University of Chicago. It's my pleasure to be here today to provide an update on the treatment of metastatic triple-negative breast cancer. It's been an exciting few years, and there are a lot of new therapies that are now available for the treatment of metastatic triple-negative breast cancer, and I'm really excited to share those data with you. So what is triple-negative breast cancer? I know a lot of you on the call are well aware of what triple-negative breast cancer is, but for any newcomers, I just wanted to go through this again. So triple-negative breast cancer is a type of breast cancer that lacks expression of the estrogen, progesterone, and HER2 receptors. Those are three receptors that we always look for any breast cancer, because we know that not all breast cancers are the same, and how breast cancers express those receptors really helps us figure out how we're going to treat different forms of breast cancer. So triple-negative breast cancer lacks expression of ER, PR, and HER2. It accounts for about 15 to 20% of all breast cancers. It's generally been observed to be more common in young women, those who are premenopausal, those individuals of African and Hispanic ancestry, those with BRCA1 associated breast cancers. So about 85 to 90% of women who have a BRCA1 germline mutation and develop breast cancer have triple-negative breast cancer. Triple-negative breast cancer historically has been associated with a poor overall survival as compared to other forms of breast cancer in the advanced stage setting. It's generally believed to be a bit more aggressive, and historically we've had limited treatment options. But as you're going to see, we have developed newer therapies for different forms of triple-negative breast cancer. Now, triple-negative breast cancer also isn't just one type of breast cancer. These breast cancers all get lumped together because they don't express the estrogen receptor, the progesterone receptor, and the HER2 receptor. But work out of Vanderbilt University has suggested that there are at least 4 distinct subtypes of triple-negative breast cancer, and potentially even more. They did an in-depth study of over 700 triple-negative breast cancers and found that these 4 distinct subtypes are driven by different genetic abnormalities. So the basal-like 1 subtype seems to be the subtype that's characterized by abnormalities in the ability to repair DNA. The basal-like 2 may be by sort of overexpression of genes that can help to drive breast cancer growth. The luminal AR subtype is a subtype that has been characterized by overexpression of the androgen receptor. The androgen receptor is a receptor that's overexpressed in a large proportion of prostate cancers. So this subtype has been treated with anti-androgen receptor therapies like those that we use to treat prostate cancer, and those have shown promise for some of these types of breast cancers. And the mesenchymal-like subtype is characterized by other features, and FGFR, which is a receptor, the fibroblast growth factor receptor, and there are some treatments that are in the clinic and in clinical trials that are targeting that. So lots of really interesting studies that are ongoing in the clinic, not quite ready for prime time, but I do want to talk a bit about some of the newer therapies that we've been able to study and get FDA approved in the last few years. So here is how we treat metastatic triple-negative breast cancer. For the most part, the treatments that we use are chemotherapy treatments, and I think you've probably all heard of many of these therapies. For the most part, we use single chemotherapy drugs at a time, but there are occasions where we might consider combinations of different chemotherapy drugs to treat advanced stage disease. I wanted to highlight, though, some of the recent approvals that we've been able to get for advanced triple-negative breast cancer. So for those individuals who have germline mutations in BRCA1 and 2, we now have PARP inhibitors that are FDA approved. And for those individuals who have PD-L1 positive triple-negative breast cancer, we now have immunotherapy that is FDA approved. And so you can see that how we treat advanced triple-negative breast cancer is dependent on whether or not individuals have BRCA1 or 2 germline mutations, and if they do, they can be candidates for PARP inhibitors. The two that are currently FDA approved are olaparib and talazoparib. If individuals have PD-L1 positive triple-negative breast cancer, then they can be eligible for treatment with immunotherapy with a drug called atezolizumab, which is an anti-PD-L1 inhibitor, and that's combined generally with chemotherapy with a drug called nab-paclitaxel. And we also have an approval that's not specific to triple-negative breast cancer, or even breast cancer at all, but really targets those cancers that have microsatellite instability or mismatch repair. Now, not very many breast cancers are characterized by abnormalities in this pathway. It's generally under 4%. But if a breast cancer does have microsatellite instability or mismatch repair, then those breast cancers can be treated with a different immunotherapy drug, an anti-PD-1 therapy called pembrolizumab. So we've studied checkpoint inhibitors or immunotherapies that target PD-1 or PD-L1 in triple-negative breast cancer. And the two largest trials looked at the anti-PD-L1 therapy atezolizumab and the anti-PD-1 therapy pembrolizumab in patients with advanced triple-negative breast cancer. And the response rates are pretty modest. So if atezolizumab or pembrolizumab were given as the first treatment for metastatic triple-negative breast cancer, the response rates were in the low to mid 20% range. But if these drugs were given in the second line setting or even after that, the response rates really dropped to under 10%. So it really does seem that these drugs are more effective when they're given as the first treatment for triple-negative breast cancer in the metastatic setting. Now, these response rates of 21 to 24% are pretty modest. And so a lot of the studies that are ongoing now or have recently been reported and published looked at combination strategies, because we want more than just 20% of patients with advanced triple-negative breast cancer to benefit from immunotherapy. So the Impassion 130 trial was the first randomized phase 3 trial that studied immunotherapy for metastatic triple-negative breast cancer that was reported and actually led to the approval of the combination of nab-paclitaxel, which is a chemotherapy drug, with an immunotherapy drug, atezolizumab, for patients with metastatic triple-negative breast cancer. This was a large randomized phase 3 trial that randomized participants to receive chemotherapy with nab-paclitaxel, with atezolizumab, the immunotherapy drug, or a placebo. There were over 900 patients who participated in this trial. And what we found was that those patients with PD-L1 positive triple-negative breast cancer actually benefited from the addition of immunotherapy to chemotherapy. So when those patients with PD-L1 positive triple-negative breast cancer got atezolizumab with chemotherapy, their cancer remained under control for about 2.5 months longer, and they lived for almost 10 months longer than those patients who got chemotherapy alone. So the findings of the Impassion 130 trial led to the FDA approval of the combination of atezolizumab and nab-paclitaxel for PD-L1 positive metastatic triple-negative breast cancer. About 40% of the patients who participated in this trial had PD-L1 positive breast cancer. And I'll say in real-world practice, it doesn't really seem to be that high. It's probably closer to 20 or 30% of patients with metastatic triple-negative breast cancer who have PD-L1 positive breast cancer, but this is now considered the standard of care treatment for PD-L1 positive metastatic triple negative breast cancer. There are a number of randomized phase 3 trials that are ongoing now looking at adding things to chemotherapy and immunotherapy. So trials looking at adding the MEK inhibitor, cobimetinib, to chemotherapy and immunotherapy, and trials looking at adding the AKT inhibitor, ipatasertib, to the same combination. Some of the initial phase 1 and 2 trials looking at the 3 drug combinations looked really promising. So it'll be really exciting to see how these randomized phase 3 trials pan out. Hopefully we'll get some results in the next year or so. And if positive, then that could continue to build on the early promise of chemotherapy and immunotherapy that we've already seen from the Impassion 130 trial. Another trial, the Keynote 355 trial, was a similar trial looking at chemotherapy with or without immunotherapy. This trial looked at the addition of the immunotherapy drug pembrolizumab, which is an anti-PD-1 inhibitor. So targeting the same pathway that atezolizumab does, but looking at, again, patients with metastatic triple negative breast cancer. This trial was a little bit different than the Impassion 130 trial. It did look at chemotherapy with a placebo versus chemotherapy plus the immunotherapy drug pembrolizumab, but this trial looked at 3 different chemotherapy backbones. Nab-paclitaxel, like the backbone that was studied in the Impassion 130 trial, or paclitaxel, or the combination of carboplatin plus gemcitabine. So patients could have received one of these 3 different chemotherapy drugs. And which drug a patient received was really based on whether or not they'd received chemotherapy before, either for early stage breast cancer, and if so, how long it had been since they'd received chemotherapy in the past. So again, chemotherapy with or without pembrolizumab, and similar to what was seen in the Impassion 130 trial, those patients who got chemotherapy plus immunotherapy did better than those patients who got chemotherapy alone. Their cancer remained under control for about 3 months longer, but only in the subset of patients who had PD-L1 positive metastatic triple negative breast cancer. Those patients who had PD-L1 negative disease really didn't seem to benefit from the addition of immunotherapy. So while this regimen is not yet FDA approved, I suspect based on this trial, we will likely see an approval of pembrolizumab with chemotherapy based on this Keynote 355 clinical trial. So hopefully another option in our armamentarium for treating metastatic triple negative breast cancer. So moving on to talk a bit about PARP inhibitors. We currently have 2 PARP inhibitors that are FDA approved for advanced triple negative breast cancer, olaparib and talazoparib. Olaparib was FDA approved based on the Olympiad randomized phase 3 trial, and talazoparib was approved based on the Embraca trial. Now, PARP inhibitors are only approved for triple negative breast cancer or hormone receptor positive breast cancer in the setting of a germline mutation in BRCA1 and 2. So to be eligible for this treatment, patients have to have a BRCA1 or 2 germline mutation. Both the Olympiad trial and the Embraca trial were pretty similar. They were randomized phase 3 trials that compared the PARP inhibitor to chemotherapy of physician's choice. Both trials showed that the PARP inhibitor actually helped to keep the cancer under control longer than chemotherapy did, in general by about 3 months. Not only were the PARP inhibitors shown to be more effective than chemotherapy, they were actually also shown to be more tolerable and were associated with a better quality of life than chemotherapy. Now, a third trial, the Brocade 3 trial, was just presented at the end of last year at the European Cancer Conference, ESMO. And this trial was a little bit different than the Olympiad and Embraca trial. This trial also was a randomized trial, but it randomized individuals with BRCA1 or 2 associated breast cancers to chemotherapy with or without the PARP inhibitor veliparib. So 2/3 of the patients got veliparib plus chemotherapy, and 1/3 of the patients got a placebo with chemotherapy. And the Brocade-3 trial, just like the Olympiad and Nebraska trials, showed that those individuals who got the PARP inhibitor actually had better outcomes. Their cancers remained under control for, on average, 6 months longer than if they had received chemotherapy alone. So a little bit of a different strategy here. The PARP inhibitor was combined with chemotherapy as opposed to being compared to chemotherapy. But again, those patients who received the PARP inhibitor did better than those patients who got chemotherapy alone. So while veliparib is not yet FDA approved, I think the manufacturers of veliparib are hoping to get FDA approval, and so that could potentially add an alternative PARP inhibitor. And again, in this setting, being combined with chemotherapy as compared to chemotherapy alone. So it'll be interesting to see how this might add an option for patients with BRCA1 and 2 associated advanced triple-negative breast cancer, and could potentially be another treatment strategy for patients with advanced stage disease. Now, approved just a few months ago is a new drug called Sacituzumab Govitecan. Sacituzumab is a drug, a class of drug called an antibody-drug conjugate, and it targets a protein called TROP2. So what is an antibody-drug conjugate? I like to think of antibody-drug conjugates as targeted chemotherapies. So basically, antibody-drug conjugates consist of 3 different regions: an antibody that targets a particular protein. In the case of sacituzumab, it targets the TROP2 protein. It's linked to a chemotherapy drug by a linker. And so the idea behind antibody-drug conjugates is that the antibody goes and binds to the protein that is on the surface of cancer cells. And when it binds to this protein, the whole antibody-drug conjugate gets swallowed up by the cancer cell, and then the chemotherapy drug is delivered inside the cancer cells and kills the cancer cells. So the idea behind these drugs is that they can provide a very high concentration of chemotherapy inside the cancer cells. So the strategy here is to really deliver a very effective therapy directly into these cancer cells. And this strategy has been shown to be effective for other types of cancers, and also for HER2-positive breast cancer, where TDM1, which is an antibody-drug conjugate, is FDA approved for advanced HER2-positive breast cancer. So sacituzumab was approved based on a small study, actually, of just under 70 patients who have had advanced triple-negative breast cancer. And what we found in this small study was that while these patients had received a number of different therapies for their triple-negative breast cancer, about 1/3 of patients had a nice response to this drug when given alone. So in the upper right-hand corner here is what we call a waterfall plot. And along the x-axis is a 0. And for those bars, and each bar here represents an individual patient, for each bar that is below the 0 on the x-axis, that means that these individual patients had their tumors shrink on this therapy. If the bars go above the 0, that means that the tumors grew. And so I think you can see here that the majority of patients actually had their cancer shrink when they got sacituzumab, and 1/3 of patients had their tumor shrink pretty significantly. So really impressive efficacy in patients who'd had a number of different treatments for their advanced triple-negative breast cancer. And based on this small study, this drug was FDA approved just a few months ago. There was a larger randomized phase 3 trial that was done called the ASCENT trial that compared sacituzumab to other chemotherapy drugs that we typically use to treat advanced triple negative breast cancer. And that trial, while we're still waiting for the results to be reported, we know from a press release that Sacituzumab was actually found to be more effective than chemotherapy. So really exciting news, and a new drug that is now FDA approved for patients with metastatic triple negative breast cancer. There are also 2 other antibody drug conjugates that are being investigated for advanced triple negative breast cancer. One is called Ladiratuzumab, and that targets a protein called LIV1A, and then Trastuzumab deruxtecan, which is an antibody drug conjugate that targets HER2 and is actually already FDA approved for HER2 positive breast cancer, but is also being studied in those patients who have triple negative breast cancer and a very low level of the HER2 protein expressed, so 1+ or 2+. And some of the initial data in the phase 1 and 2 settings for both of these drugs are showing promise for subsets of patients with advanced triple negative breast cancer. So hopefully in the not too distant future, we'll be hearing more about those drugs and how effective they are in triple negative breast cancer. I'd like to end just by talking a bit about the androgen receptor positive subset of triple negative breast cancer. So AR positive triple negative breast cancer accounts for about 10 to 15% of triple negative breast cancers. And there have been a number of studies looking at anti-androgen receptor drugs, enzalutamide, abiraterone, bicalutamide for AR positive triple negative breast cancer. Now, all of these drugs, bicalutamide, enzalutamide, and abiraterone, are already FDA approved for the treatment of prostate cancer, because the vast majority of prostate cancers actually express the androgen receptor. And so we're hoping to repurpose some of these drugs for the treatment of AR positive triple negative breast cancer. One of the largest studies looked at enzalutamide in patients with AR positive triple negative breast cancer. And what we found was that the higher levels of androgen receptor positivity, so if 10% or more of the breast cancer cells in a triple negative breast cancer patient express the androgen receptor, those patients actually did better than lower levels of androgen receptor expressing triple negative breast cancers. So, a lot of interesting early modest efficacy for some of these agents. But unfortunately, the development of these agents has been stalled in the past few years. There is a trial, however, that's going to be starting up in the not too distant future, hopefully in the next few months, looking at some of these drugs, specifically enzalutamide, in AR positive metastatic triple negative breast cancer. This trial is going to be led by Dr. Tiffany Traina and myself, and will be run through the Translational Breast Cancer Research Consortium. And so this trial is looking at comparing chemotherapy, which is how we treat androgen receptor positive triple negative breast cancer now, comparing chemotherapy to enzalutamide plus a second drug called mifepristone. Mifepristone is a glucocorticoid receptor blocker, and it's been shown that glucocorticoid receptor activity can actually lead to resistance to enzalutamide. So the idea behind adding mifepristone here is to see if adding mifepristone to enzalutamide might help increase the efficacy above and beyond enzalutamide alone, and then comparing both of these sort of targeted strategies to standard of care chemotherapy, with the hopes that we can develop better treatments for androgen receptor positive metastatic triple negative breast cancer. So I just wanted to end by recapping what I've just talked about. And as I'd mentioned before, a lot of excitement, a lot of exciting new therapies that are now FDA approved for metastatic triple negative breast cancer. Immunotherapy, we first started studying back in 2013, and now immunotherapy is FDA approved for metastatic triple negative breast cancer that expresses the PD-L1 protein. It's been, immunotherapy's been studied by itself, but really adding chemotherapy seems to enhance the effectiveness and help more patients benefit from immunotherapy. But there are a number of trials that are ongoing looking to add a third drug, either a MEK inhibitor or an AKT inhibitor, to chemotherapy and immunotherapy to see if we might be able to help even more patients with metastatic triple-negative breast cancer benefit from immunotherapy-based strategies. PARP inhibitors are already approved for BRCA1 and 2 associated metastatic triple-negative breast cancers. There are a whole set of studies now ongoing looking at adding immunotherapy to PARP inhibitors to see if that might be an effective strategy. Antibody drug conjugates are showing a lot of promise. Very exciting to see the first antibody drug conjugate approved for metastatic triple-negative breast cancer just a few months ago. The drug Sacituzumab Govitecan, which is a really effective treatment that is approved for those patients who previously received at least 2 therapies for their metastatic triple-negative breast cancer. And it'll be really interesting to see, hopefully in the not too distant future, maybe later this year at San Antonio, the results of the large randomized phase 3 trial. And not to be left out, targeting the androgen receptor in AR-positive triple-negative breast cancer has shown early promise. And there is a Translational Breast Cancer Research Consortium trial that will be hopefully opening in the next several months at many sites across the country. So a lot of exciting targeted therapies that are continuing to build on the success and promise of chemotherapy for metastatic triple-negative breast cancer. It's been my pleasure to be here today to talk to you about what's going on for triple-negative breast cancer, and I look forward to answering any questions that you have. Thank you for your time.